Hello everyone. So this is the last part of our chapter, which is photosynthesis in higher plants. So till now we have covered a portion till yes, we have covered it. Yeah, we have discussed this chemosmotic hypothesis also, and we are going to discuss that maybe from here that where the ATP or no, I guess this is also done. We are going to discuss about the Calvin cycle, I guess. So let's discuss about it. So what is a Calvin cycle? A Calvin cycle and his Calvin and his co-workers then tried to work out the whole pathway and showed that the pathway operated in a cyclic manner. IUBP was generated. IUBP was generated, and how the Kelvin pathway operates we are going to discuss that how it happens. And let us now understand that it is very clearly that the Calvin pathway occurs in all the photosynthetic plants, means the plants which have chlorophyll, right? Okay, next is written that it doesn't matter whether the plant is following the C3 pathway or the C4 pathway. It is important, or any other pathway, but it is important that all the photosynthetic plants will go through the pathways. Okay, whether it is C3 or C4 or any other pathway. Okay, let's talk about the three stages of Calvin cycle, which are carboxylation, reduction, and regeneration. So let's talk about it. Carboxylation. It is the first step of our Calvin cycle. So what exactly happens in the carboxylation? Let's discuss. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is most crucial step of the Calvin cycle where the CO2 is utilized. CO2 is utilized for the carboxylation of IUBP, and this reaction have two molecules of 3-PGA. Since the enzyme which is used is an oxygenation, and so is this process is called as IUBP carboxylase oxygenase or rubisco. Why we are calling it rubisco, or we are calling it because look what happened. So in this process carboxylation is done, and with the carboxylation there was another step which is oxygenation. Therefore we call it carboxylase oxygenase, or we can say we call it rubisco. Okay, so let's talk about the next step. What happens in the next step? Next step, look, we are going to first discuss about this. No, first we are going to read. So what is it saying? It is saying that second step is reduction. What happens in this process? It is a series of reaction that will lead to the formation of what? That will lead to the formation of glucose. A glucose. C12H24O11. Okay, it will lead to the formation of our glucose, and the steps involved is the utilization of two molecules of ATP, two molecules of ATP for the phosphorylation with the 2 of NADPH, 2 of ATP, 2 of NADPH. Okay, and for the reduction per CO2 molecule fixed. Okay, so the fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of 1 molecule of glucose. Okay, and third step we have is regeneration. Regeneration of the CO2 acceptor molecule IUBP is crucial to continue, and the cycle will not interrupt in between, and the regeneration step requires 1 ATP for the phosphorylation to make IUBP. So we need 1 ATP for the phosphorylation, so that it can lead to the formation of IUBP. Okay, and let's discuss about the diagram. So what is happening in this diagram? There we have first step, which is carboxylation. We have ribulose 1,5 bisphosphate. From the atmosphere we'll get CO2 and H2O. Carboxylase, then we're going to make 3-phosphoglycerate from it. Next what will happen? Look, next reduction step will be going to take place in reduction. We form ATP or phosphate plus NADP from ATP and NADPH. Then triose phosphate will make, and we also have something here. It is sucrose and starch. What is it? Sucrose and starch. Next, what will happen? It will go to the last step, which is regeneration. Regeneration also ATP will going to make ADP. Okay, and last this cycle will continue. So this is also a cyclic process, as it is working is in a cyclic manner, and it is endless. Endless. Okay. Okay, so let's move on to next. Henceforth, every CO2. Look, this is very important. This is very important. This is saying that what goes inside and leads to the formation of which substance. Okay, so six CO2 molecule will give one glucose. This is very important. Six CO2. Write it down somewhere. CO2 will give one glucose. Okay. 18 ATP will give 18 ATP. This is same. 12 NADPH will give 12 NADP. This is also same. You need to learn that 6 CO2 leads to the formation of one molecule of glucose. Let's talk about the C4 pathway. Plants have the adaptation. Like, you must be thinking, when we have C3 pathway, then what is the need to we have C4 pathway? No. So if all the photosynthetic plants are going to do the same, so why can't they do the C3? Why can't they follow the C3 pathway? It is because the C4 plants have some different adaptation. Because of the adaptations, they have different pathways. Okay, so according to the adaptation, this is differentiated. So the plants that are adapted to dry tropical regions have the C4 pathways, and because of their adaptation only they have the difference. Like, from the C3 plants, they are different because they are following the C4 pathway because of their adaptation only. Okay, so these plants have C4 carboxylation acid as their first fixation product, and they use C3 pathway Calvin cycle as the as it is mentioned. And what happens in the C4 plants are why they are special. They are special because of their leaf anatomy. They have a different kind of anatomy. They have a special type of leaf anatomy. Okay, because of their special kind of leaf anatomy, they are special from others, and because of it only they can tolerate the high temperature, and they can respond to high light intensity, and this process called as photorespiration, and this leads to a greater productivity as compared to C3. The greater productivity will have the C4 plants. Okay, and the reason is very much clear. They have this special kind of leaf anatomy, therefore they are adapted to the dry tropical region, hence they can tolerate the high temperature, they can respond to high light intensity, and this process is, we call it, is photorespiration, and they have the greater productivity as we are comparing it with C3 pathway. So how this has happened? So the particularly large cells around the vascular bundles of C4 plants are called bundle sheath cells, and the leaf that has such kind of anatomy, which is the special kind of anatomy we discussed earlier, before, right? So it is Kranz anatomy. What is it? Kranz means breadth, and we can say it is a reflection of arrangement of cells, that how the cells are arranged, and due to which what kind of what kind of anatomy is formed. What is the anatomy, right? So let's talk about it. So they have a large number of chloroplasts. They have thick wall which is impervious, which resist, which means resist or doesn't allows the gaseous exchange of gases, and no intercellular spaces they have. And when you cut it, like, you can cut the maize and sorghum to understand what kind of Kranz anatomy they have. Okay, so it would be interesting for you to collect the okay, this you will read only. Now let's study that how the pathways work. So this pathway has been named on the Hatch and the Slack. This is called as Hatch and Slack pathway, and again it is a cyclic process. It is somehow similar to C3, but a very differentiated form of C3 pathway. Okay, it is kind of different. Okay, let's talk about it. So the primary CO2 acceptor is 3 carbon molecule, which is phosphophenol pyruvate, which we call commonly PEP. Okay, and it is present in the mesophyll cells. The enzyme which is responsible for its fixation is called as PEPcase. Very simple to learn it, because PEP का fixation कौन करवाएगा? PEPcase ही करवाएगा, right? So PEPcase भी कह सकते हैं, PEP carboxylase भी कह सकते हैं. It is important to register that the mesophyll cells lack the rubisco. They don't have this rubisco that we earlier discussed, no? So they don't have that enzyme which is rubisco. Okay, next is saying that the C4 acid OAA is oxaloacetic acid. Okay, oxaloacetic acid. So it is formed in the mesophyll cells. So in the forms other four carbon compounds like malic acid or aspartic acid is present in the mesophyll cells, okay, which are transported to the bundle sheath cells. Then in the bundle sheath cell, what happened? These C4 acids are broken down to release CO2 and 3 carbon molecule. The 3 carbon molecule is then again transported back to the mesophyll, where it is converted to PEP again, and this is how the complete cycle works, and this will continue as it is a cyclic process. Okay, so look here, what is it happening? This is the mesophyll cell. Okay, and here we, from the atmosphere, we take the CO2. There it will lead to the phosphophenol pyruvate fixation, C4 acid transported back again, and here it will form the C4 acid, decarboxylation of CO. When CO2 releases, then it is transported back, and this process will continue. The first with C is happening in the mesophyll cells, and second it is happening in the bundle sheath cells. Okay, next we have the enzyme ribulose bisphosphate carboxylase, which is none other than rubisco. Okay, next we are going to talk about photophosphorylation. So what is phosphorylation? It is difference. It creates an important difference between the C3 and the C4 plants, which is phosphorylation. To understand, we need to learn that the first step of Calvin cycle pathway is the first CO2 fixation. Then this is the reaction where the RUBP combines with CO2 to form two molecules of 3-PGA and catalyzed by enzyme, which is very famous, rubisco. Okay. Next, RUBP plus CO2 in the presence of rubisco enzyme will give two molecules of 3-PGA. Okay. Rubisco is most abundant enzyme in the world. You need to learn about it. Okay. And rubisco has much higher affinity for CO2. It somehow likes to bind with CO2, okay, more than O2. Okay, so that's the problem of rubisco, that it has greater affinity for CO2. And imagine what will happen when, okay. In C3 plants, O2 doesn't bind with rubisco, hence the CO2 fixation is decreased. RUBP, instead of being converted to two molecules, binds with O2 to form one molecule of phosphoglycerate, and phosphoglycerate is the path which we call as photophosphorylation. For photorespiratory pathway, there is neither synthesis of sugar nor of ATP, rather it rests through the release of CO2 only. Okay, CO2 with the utilization of ATP. Do you know that why in the C4 plants photophosphorylation doesn't occur? This is because they have the mechanism that increases the concentration of CO2 at the enzyme site, or it takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cell to release CO2. Only this is result in the increasing intercellular concentration of O2. In turn, this enzyme that rubisco function as carboxylase minimize the oxygenase activity. Okay, so that's why it is not happen in the C4 pathway plants. Next, we are going to discuss about this factors affecting photosynthesis. So what are the some factors which affects or which somehow decrease the effect for photosynthesis. Okay, so we are going to discuss about it. So photosynthesis is necessary. Okay, it is necessary, and the rate of photosynthesis is varies from different, like it depends on the situation, or determine it somehow determines the plants, including crop plant. Photosynthesis under the influence of several factors, whether it is external or internal. So the factors includes number, size, age, orientation of leaves, and etc, and etc. Mesophyll cells, chloroplast, internal CO2 concentration, amount of chlorophyll, etc. Okay, so the external factors include the availability of sunlight, that how much the sunlight is available for it or not. Temperature also matters, that how is the temperature, because it affects. Okay, CO2 concentration, whether the concentration is high or low, whether water is supplied or not. So as a plant photosynthesis, all these factors will simultaneously affect its rate. Like its rate will get affected by all of these external plus internal factors. Okay, so several factors interact simultaneously affect the photosynthesis. Okay, so what are these factors? Let's talk about them only. Okay, this is something very important. When several factors affect any biochemical process, Blackman's in 1905 gave the law of limiting factor, which says that if a chemical process is affected by more than one factor, then its rate will determined by the factor which is nearest to its minimal value. So this law is some very important. This law is very important, which says that if in any chemical reaction, if there is something which is getting affected by more than one factor, then the rate will determine by the factor which is nearest to its minimal value, which is nearest to its minimal value. This will affect the rate of it. First we are going to discuss about the light. So light quality, light intensity, duration of exposure, that how much time the sunlight is given, or we can say light energy is given to it, or this is a factor which affects the photosynthesis, and it has a linear relationship with the rate of photosynthesis. Okay, at higher light intensities the rate doesn't show further increase as other factor becomes limiting. Next we have CO2 concentration. CO2 is the major limiting factor for photosynthesis. Concentration of CO2 is low in the atmosphere between 0.03 and 0.04%. Okay, and the increase in the concentration up to 0.05% can cause to increase in CO2 fixation rates, and this level will become damaging and low in longer periods. So C3 and C4 plants respond differently to CO2 concentration. So at low light condition, neither group respond to high CO2 conditions. Okay, so this is a factor which will affect it, and there are some values that have given, you can read it from the book. Next we have temperature. Dark reactions are carried out by the enzymes, are temperature controlled, and though the light reaction are also temperature sensitive, they are affected to a much higher extent. So the C4 plants respond to higher temperature and show higher rate of photosynthesis, while C3 have much lower than C4. The temperature option is much lower. Okay, next we are going to talk about the water is the last maybe. And even though water is one of the reactant in the light reaction, it is a factor which affects the rate of photosynthesis rather than directly on photosynthesis. Water stress causes what happens, like this is a stomata. Due to water stress, the stomata will get closed, and then this will reduce the availability of CO2 will get reduced, and therefore the water stress can also make the wilt leaves wilt. This reduce the surface area of the leaves and metabolic activities as well, and this overall leads to affect the rate of photosynthesis. Okay, so we are done with the chapter. So like, share and subscribe to my channel for more videos. Thank you very much.